Welcome to my channel XY Biology Academy. In natural environments, microorganisms rarely occur as single species. Instead, they exist as mixed populations containing numerous types of microorganisms. For the accurate study of an organism's cultural, morphological, physiological, and biochemical characteristics, it must first be isolated in the form of a pure culture. The streak plate method is one of the most widely used microbiological techniques for obtaining pure cultures. It is a mechanical dilution technique in which a small amount of mixed culture is progressively spread over the surface of a solid agar medium. Sterilization of the inoculating loop between successive streaks progressively reduces the number of bacterial cells transferred to each new section of the plate. This gradual dilution separates individual bacterial cells from one another across the agar surface. Materials required: mixed bacterial culture, culture media, sterile inoculating loop, Bunsen burner or micro-incinerator, marker or laboratory marking pencil, test tube rack, incubator. Procedure. Step 1. Labeling the plate. Label the bottom surface of the sterile agar plate with the experiment title, date, sample identification, and initials. Always label the base of the Petri plate rather than the lid to prevent misidentification if the lid is accidentally removed. Step 2. Sterilization of the inoculating loop. Sterilize the inoculating loop by heating it in the flame of a Bunsen burner or micro-incinerator until the wire becomes red hot. Allow the loop to cool by gently touching an unused sterile portion of the agar near the edge of the plate. A hot loop should never be used to collect bacterial culture, as it may destroy the microorganisms. Step 3. Collection of the inoculum. Flame the mouth of the culture tube, insert the cooled sterile loop into the broth culture, and collect a loopful of the mixed bacterial suspension. Flame the mouth of the tube once again before replacing the cap. Step 4. Streaking the first quadrant. Partially lift the lid of the Petri plate and streak the inoculum over area 1 using closely spaced parallel streaks. This first quadrant contains the highest concentration of bacterial cells and serves as the starting point for subsequent dilution. Step 5. Sterilization before the second streak. Sterilize the inoculating loop until it becomes red hot, allow it to cool, and rotate the Petri plate approximately 90 degrees. Step 6. Streaking the second quadrant. Touch the cooled sterile loop once at the edge of area 1 and drag the bacteria into area 2 using several parallel streaks. Once the streaking of area 2 has begun, do not return the loop to area 1. Step 7. Sterilization before the third streak. Sterilize and cool the inoculating loop again. Rotate the Petri plate another 90 degrees. Step 8. Streaking the third quadrant. Touch the edge of area 2 once and streak the bacteria throughout area 3. At this stage, fewer bacterial cells are transferred, resulting in a greater degree of dilution. Step 9. Streaking the fourth quadrant. Rotate the Petri plate another 90 degrees. Following the standard 4-quadrant streak method, drag the bacteria from one corner of area 3 into area 4 using a wider streaking pattern. Avoid touching any previously streaked areas except at the initial point of contact. This final streak provides the greatest dilution and facilitates the development of well-isolated colonies. Step 10. Incubation. Close the Petri plate, invert it to prevent condensation from dripping onto the agar surface, and incubate the plate at 37 degrees Celsius for 48 to 72 hours. Observation. After incubation, examine the agar plate carefully and observe the distribution of bacterial growth. Typically, heavy confluent growth is observed in area 1, moderate growth in area 2, lighter growth in area 3, and well-isolated discrete colonies in area 4. In this experiment, we will learn the spread plate technique, a commonly used microbiological method for inoculating microorganisms on the surface of a solid agar medium. The spread plate technique is based on the principle that a previously diluted microbial suspension is evenly distributed over the surface of a solid agar medium using a sterile L-shaped glass spreader. Materials required for this practical include the following: sterile nutrient agar plate, Bunsen burner, Marker for labeling. Sterile inoculating loop, diluted culture of bacteria, sterile water, an L-shaped glass spreader. 95% ethyl alcohol, a beaker, and a turntable. Now let's perform the practical step by step. Step 1. Sterilizing the glass spreader. Take a beaker containing 95% ethyl alcohol and immerse the bent portion of the L-shaped glass spreader into the alcohol. This helps sterilize the spreader before use. Step 2. Preparing the agar plate. Take a sterile nutrient agar plate and label it properly with the required information. Place 1 drop of sterile water in the center of the agar surface. Then, using a sterile inoculating loop, transfer a loopful of the previously diluted bacterial culture into the water drop and mix it gently. Step 3. Flame sterilization. Remove the glass spreader from the alcohol and carefully pass it through the flame of the Bunsen burner. Keep the bent end of the spreader pointed downward while flaming to prevent burning alcohol from flowing toward your hand. Allow the alcohol to burn off completely, then let the spreader cool for about 10 to 15 seconds. Never use a hot spreader directly on the agar surface, as it may kill the microorganisms or damage the agar. Step 4. Spreading the inoculum. Slightly lift the lid of the Petri plate and place it on the turntable. A turntable in the spread plate technique is a flat rotating platform on which the Petri dish is placed. As it spins, the L-shaped glass spreader remains in contact with the agar surface, allowing the microbial suspension to spread evenly. While the plate is rotating, gently touch the cooled spreader to the agar surface and move it smoothly back and forth. Continue spreading until the microbial suspension is distributed evenly across the entire surface of the agar. Step 5. Completing the inoculation. Once the culture has been spread uniformly, replace the lid of the Petri plate. Immerse the glass spreader back into the alcohol and flame sterilize it again before storing it or using it for another sample. Alternative method. If a turntable is not available, the same procedure can be performed by rotating the Petri plate manually while gently moving the sterile glass spreader over the agar surface. After incubation, the microbial cells grow only on the surface of the nutrient agar, producing well-isolated colonies. These colonies can be used for identification, viable cell counting, and further microbiological studies. Now, let's learn another important microbiological method known as the pour plate technique. Unlike the spread plate method, the pour plate technique allows microorganisms to grow both on the surface and within the agar medium, making it especially useful for bacterial enumeration and obtaining isolated colonies. The procedure begins by preparing a serial dilution of the mixed culture using a sterile pipette or inoculating loop. This step reduces the number of microorganisms in the sample, making colony counting more accurate. Next, a measured volume of the diluted culture is transferred into a sterile Petri dish. Molten nutrient agar, cooled to approximately 45 to 50 degrees Celsius, is then poured into the plate. The plate is gently swirled in a circular motion to mix the inoculum evenly with the molten agar. Once mixed, the agar is allowed to solidify at room temperature. After incubation, bacterial colonies develop both within the agar and on its surface, allowing accurate enumeration and isolation of microorganisms. Here you can see the detailed comparison of various plating techniques. Thanks for watching, like this video and subscribe to my channel.